So as part of my conflict of interest management with my institution, I have to acknowledge that I do speak to people and serve on different advisory boards and do some research in my laboratory. Some of the work I'm going to talk about is from UBVac, which is a vaccine and which was a spin out of the institute and which I am a co-founder. So the outline of what I wanted to talk with you this afternoon is something about what part of the immune system cures cancer, combo or combination immunotherapy, and FDA objectives that were released in 2017. And I want to talk about a new class of cancer antigens that are these short-lived proteins that are presented, maybe the dominant epitopes in class 1 molecules. Then I want to talk about some to address both the FDA and to really improve and find out how to cure patients, I think more effectively. I want to talk about some monitoring and some novel combination immunotherapy that we have entering the clinic. So immunotherapy can cure patients with cancer. It can cure patients with cancer. And we know that because of these data. And that is because patients that achieved a complete response of high-dose IL-2, they went on to have not 20 months, but 20 years progression-free survival. So 90% of patients went out. And the woman, Linda Taylor, who was treated when I was a fellow, started treatment in the middle 80s back at the NCI. It remains disease-free last follow-up, which was April of last year. And that was somebody who had wildly metastatic melanoma. So IL-2, interleukin-2 is a T-cell growth factor. It activates T-cells and NK cells. It induces cytokine storm, which I think impacts antigen-presenting cells, innate immunity, and tumor cells. So what is the mechanism of action for cure? I would suggest we still don't know what it is. Do we know in terms of anti-PD-1, so the big checkpoint blockade, for which the Nobel Prize was given in CTLA-4, will that give us the same result? It's too early to know. We're not going to know that for another 7 to 10 years. So in 2019, the MOA, or the mechanism of action for cure, is unknown. Without knowing this, I think it's going to be difficult to perform relevant immunoprofiling, and one of my slides that I put in dropped off, but it's going to make it difficult then to improve on what we're doing. So this is an outdated slide, but there are a couple thousand clinical trials going on, combining checkpoint blockades with almost every kind of agent under the sun. Many of those are marriages of convenience of pharmaceutical companies, and they're done like this. And that is that a patient goes on the trial, the patient 12 weeks later, 8 weeks later, 16 weeks later has a scan, the patient has progressed, the patient has remained disease-free or has not had progression or has had complete regression or partial regression of their tumor. But they're not followed, in the vast majority of those studies, there's no interrogation of that patient to understand what is happening to the patient's immune system during that time interval. So the FDA in July 20th, 2017, it's all on the website, video, it's a joint FDA-AACR program that I participated in. The FDA outlined a series, I think, of 8 objectives, and I present 4 of them here. One was to identify best practices regarding patient selection for immuno-oncology combinations, to identify best practices regarding dose selection and optimization for immunotherapy, to discuss the utility of biomarkers as pharmacodynamic endpoints. And then the last one is really a complicated one, but if you're going to give somebody combination immunotherapy, you're going to give them 2 or 3 or 4 things. You got to find a way to understand how each one of those individual agents is impacting the immune system and confirm that it has that effect or doesn't have that effect, so you can better understand what's going on. So again, these are just 4 of the FDA objectives. So how are you going to do all that? I just put this slide in there. There's a lot that's been written on this. The Society for Immunotherapy of Cancer has put together a series of white papers to discuss all these in detail. I'm just going to talk about one, which is this multicolor IHC and Immunoscore. And I put these slides in after your talk this morning to talk about this just a little bit. And so Society for Immunotherapy of Cancer led a study to look across 13 countries, look at about 3,500 patients, to see whether or not this idea that immune infiltrates correlated as a prognostic biomarker in colon cancer. And that was whether or not CD8 T cells got into the tumor or were on the invasive margin of the tumor, to enumerate those and look globally, Asia, North America, Europe, and a little bit in India, whether or not that correlated with outcome. And so the results of that study were published in Lancet last May. And essentially, in that whole overview, this is an overview of that, there was a highly significant correlation between this infiltrate in this global population and as a prognostic biomarker. So I just want to say that it was important. I'm going to jump ahead now to this part. So if I can just turn the lights on a little bit, if that's possible. So this is a slide of head and neck cancer. Over here, this is where the cancer is, and then these yellow cells are CD8 T cells. So the red on the big blue dappy nuclei is PD-L1. If you take a needle and put a needle through this biopsy, you're going to get tumor cell markers, you're going to get PD-L1 expression, you're going to get CD8 expression, and oh yeah, these green cells, which are on that margin, those are T regulatory cells. So you'd get all that information from your genetic signature, but you would miss what I think is one of the most important components, which are the relationships of where those cells are spatially in terms of interacting with one another. So in the discussion we had on the questions, that's why I went back to this, because we can get a lot of data from the TCGA and looking at genomically evaluating data, but we can't get this. This is still important, and we need to figure out ways to do this. So I'll show you why it's important. So this is work that was done by a very bright MD PhD student in my lab who's currently at Baylor doing his head and neck, Carmen Ballesteros Marino, who's a scientist in the group. And anything that I say is correct is Carlo Bifulco, who's my pathology colleague who oversees all this. So we've known for a long time that if you've got high levels of CD8s in head and neck cancer, you do better. And that's what we did. This is just looking, you got high CD8s in the tumor, high CD8s in the stroma, patients do better. Look at the p-values, 0.01, 0.23. FOXP3, these are the regulatory cells, the cells that suppress the immune system. There are at least 3 other reports that show if you've got high T regulatory cells, you do better in head and neck. To a tumor immunologist, that makes no sense. That can't be true, right? But that's what it says. And this data shows, again, it's not statistically significant, but other studies are statistically significant, and that's with both counting by IHC as well as genomic profiling. PD-L1, these are patients, HPV negative head and neck cancer patients before the era of immunotherapy got standard therapy. PD-L1 expression had no impact on the outcome of these patients just looking at PD-L1 expression at the tumor or the stroma. So now, we did these studies using multiplex IHC where we had spatial information for every cell. So we had the cartographic coordinates for every cell in the tissue and could look at relationships of every cell to every other cell. So what we did is we took and focused on the CD8 cells. So you heard about that from Professor Greenberg earlier, about those are the effector cells, and well, you do that, but you look at around that CD8 cell, how many FOXP3 or how many T regulatory cells are around it, and how many PD-L1 cells are around it. And so now if you take and you look at, let's split them, the patients who have got high levels of FOXP3 around their CD8s, what you see is now it's reversed of what I showed you earlier. So high levels of FOXP3s around the CD8s, they do worse. Low, they do better. And that's true for the stroma and the tumor. Stroma and tumor here. And the p-values are now 0.005 or 0.024. PD-L1, which had no impact before, okay, now PD-L1 expression, if it's near CD8s, it does better than highly significant. So why is that? T regulatory cells and PD-L1, at least for T regulatory cells, more than half of their suppressive functions are contact dependent. They've got to be able to get next to the CD8 cell. So if they're distant from it or not related, they can't suppress. PD-L1 the same, right? PD-L1 has to bind to PD-1 to send that negative signal to the T cell and shut it down. So now we're looking at the tumor and the stroma, 2 different areas. So you should be able to take these data. So if you're low here and low here, you should do the best. If you're high and high, you should do the worst. And if you're low with one and high in the other, you should be intermediate. That's what we were thinking. And in fact, that's what turned out to be the case. So now you're also looking at 2 different areas, the tumor and the stroma. We should be able to add those together to see what happens in terms of power. And so what we did is we did exactly that. So if you're low for everything, low for FOXP3 and PD-L1 in the tumor, low for those in the stroma, you'd have 0. If you're high for all of them, you're here. And what you see is that you see a separation now. You start to see separation of curves. These haven't been weighted or any data. They're just looking at it. So if you're high for everything at 4, you do the worst. If you're the low for everything, you do the best. So I touched about 2 mechanisms two suppressive mechanisms. But there's lots of other mechanisms, right? And one of the ones that I think, again, Dr. Greenberg talked about briefly was, loss of antigen presentation. But so one of the ways we looked at that was in collaboration with Barbara Seliger, who's a professor at Halle, Germany, and a young pathology resident who came and spent some time in the lab in Portland, and looked at whether or not we had real problems looking at membrane-bound class 1. So we ended up looking at accumulation of cytoplasmic antigen presentation components, which have been associated with reduced class 1 expression and antigen presentation. So when you start and take that data now and overlay that with the data I just presented, you can now take that cohort of patients and take the patients at 0, where you've got now 60 months, so 5-year overall survival is 100%, and you identify the patients that are going to do very badly and are going to all be dead by 36 months. So this is, I think, the power of this, and this just is a prognostic biomarker. So this is a start, but this is kind of information, if you're going to enroll patients on a clinical trial, you sure should like to know if your patient you're enrolling is down here or at the top, and going to do well regardless of what you do for him or her. So that was prognostic biomarkers. Let's talk about predictive biomarkers. Spatial relationships can be predictive. And we're talking about now for checkpoint blockade. So and there's more data coming. This was from a online first publication from last year that if T cells, if you see PD-1 and PD-L1 next to each other, if they're in proximity, that's a much better predictive biomarker that they're going to respond to checkpoint blockade than just PD-L1. Because it tells you that there are T cells at the tumor site and that they're being inhibited by PD-L1. Another surrogate for that is this IDO HLA-DR expression. So if you see IDO and HLA-DR next to it, it's another surrogate probably for T cell. There are more coming. And I stuck in this one slide too, after thinking about it at lunch, which is some work really, my colleague Andy Weinberg really led this project, and Thomas Duhan is a recent addition to our faculty in Portland, looked at expression of CD39 and 103, co-expression on those cells in the TIL, in the tumor-infiltrating lymphocytes, were associated with the number of tumor-reactive T cells they were. So we think this is a marker of tumor-reactive T cells. So if you want to get a feeling, so if you've got high levels of those, and this is looking at blue, double positive blue, these patients do much better than the patients who are in the lower 50%. This is for all head and neck, and this is for HPV negative head and neck. So I guess the reason I stuck this slide in is now we've got a static marker, right? If you've got a section, if you're going to do some kind of expression, either 10x genomics looking at single cell or do something else on your multiplex IHC to figure out what's going on, you can look at these sorts of cells and say, okay, I want to take this and I want to look at digital spatial profiling. I want to know what's going on in the areas where those cells are or where they are not. This is another collaboration that I thought about when we talked about it in the lunch line today. To stick this in is just that I think this is going to be really powerful to help us, not just in the patients where we've got lots of those cells, but when you look at these tumors that are devoid of immune cells, devoid of macrophages, or have got lots of macrophages, but you can now start to focus in on there and take a little circle. You can be guided by your multiplex IHC, then take the next slide and focus on those areas and suck off the 40 proteins and the 1,600 genes or whatever it is that you're going to be able to look at in terms of transcripts to try and get some insight. And so we have a collaboration. My lab is a collaboration with Definians to do the multiplex IHC, and then with NanoString to do the digital spatial profiling, to look at mesothelioma, to try and understand that disease, what's going on. The reason we picked that is because we did a whole cohort of mesothelioma patients, and then just when we were ready to publish it, in the ESMO journal came out a paper which was exactly the opposite of what we found in terms of with multiplex IHC. So we didn't publish our data. We want to kind of get into it a little bit and see what's going on in that disease. So mutations, mutational burden, high TMB, that's a predictive biomarker, or maybe a prognostic biomarker depending on the disease. This is a really interesting paper out of Tom Gajewski's lab. It was head by Stephanie Spranger, who's now at the Koch Institute at MIT in Boston. But if you focus on one, let's pick melanoma. What's really interesting here, and so this is looking at the number of non-synonymous mutations, so the mutational burden of melanoma. And if you're looking at red, this is the tumors that are inflamed, and blue are the tumors that are not inflamed. And why I show you this is it looks like there's an equal distribution. It doesn't make any difference what the mutational burden or the number of immunogenic antigens potentially that are there. There's something else completely unrelated. Well, maybe it's related to this, but the point is that you can have both a hot tumor and a cold tumor at all the spectrum, even at the least mutated cancers, as well as at the highly mutated cancers. But it doesn't mean they're going to be inflamed. We've heard about this over again. Everybody knows this. Cancer is heterogeneous. We know about this immunoediting component, right? That the immune system can eliminate clones, but clones can grow out that are going to now be able to escape the immune system because of loss of class 1 or loss of a specific antigen. So does that happen with PD-1? And in fact, this paper that was published in Cell now more than a year ago looked at that. So what you're looking at here really quickly is you're looking at response to therapy, you're looking at the tumor biopsy, and this is just the cartoon from the Cell paper. But essentially you've got tumor cells here, and they express either red neoantigens, green neoantigens, or black neoantigens. And when the patient gets, and these are the T cell clones, when the patient gets anti-PD-1, this patient lost these 3 tumor cells, which all have the black neoantigen on them, and they had, at the same time, they had an expansion of their T cell clones, which were probably recognizing the neoantigen. This is all kind of hypothetical, but it fits with these concepts. So this is what's going on. So I think part of the idea that we have is you want to be able to look more broadly at developing not just T cells that are going to recognize these other epitopes, but you want to do things to hit your iNKTs so you can actually get those as well as your NKs. And let's not forget about B cell immunity and immunoglobulins that may both give you ADCC with NK cells or complement and other types of effector mechanisms. So this paper is a little bit of a red herring. He doesn't really talk about the immune system so much, but it's mentioned a little bit in there. So the search for cancer treatment beyond mutant hunting. I think this is going to be critically important for the vast majority or a large majority of cancer patients, because there are many patients who are going to have low mutational tumor burden. And so there are lots of other opportunities. And this is a paper, this first one is one that the NCI ran, and actually Mac Cheever spearheaded this effort from the NCI to prioritize cancer antigens. And more than 60 of those are things that are not mutated epitopes. And one of them is mesothelin, which you heard Professor Greenberg mention earlier today. But I also see the Cancer Genome Atlas as being an incredible reservoir of information for us as immunologists to understand what are the targets we should be looking at. So they've identified the genes in cancer that are upregulated and amplified compared to normal tissue. And we also give you associations with survival. And I'll show you how we're using some of that data to try and interpret responses in patients who are receiving immunotherapy at the Chiles Research Institute. This slide disappeared. I don't know, this is Mac to Mac, but something happened. So this paper out of Mark Davis and Christopher Garcia's lab was published in Cell. And what they did is and I'll just do it really quickly, they were looking at T cells in 2 colon cancer patients. And what they found was that 3 of these 4 T cell receptors recognized non-mutated peptide that was overexpressed in colon cancer and other cancers. And they identified a shared non-mutated tumor antigen between 2 patients. And that was this U2AF2, which is overexpressed in many cancers, including colon adenocarcinoma, which is what these patients had, but also in non-small cell lung cancer, breast, and lymphoma. And this protein, U2AF2, is a protein that we've got in the vaccine that I'm going to talk to you about in a minute. It's one of the many proteins. So I think the whole point of showing you these data was to not give up on the non-mutated epitopes and on patients who've got low tumor mutational burden. So I want to talk to you kind of quickly about this slips and drips. So there's 2 different classes of proteins. There are proteins that are slips and drips, which are short-lived proteins and defective ribosomal products. This is misfolded proteins. These are the proteins that get degraded through the proteasome, pumped into the endoplasmic reticulum, and then loaded onto class 1. And these are thought to be the dominant epitopes that are expressed on the surface of all cells. And so tumor cells, and this is controlled a little bit by the number of the transcripts that you have and protein synthesis and all that. But it's really the work that Jonathan Udell at the NCI has been working on for more than 20 years. The other proteins are these long-lived proteins that are degraded through autophagy. So I'm going to skip through a couple slides here. So we've got a vaccine that is concentrated the short-lived proteins. And so this is the first publication of that work going back now a decade ago more than a decade ago, in the 3LL model. And this work that was really led by Hongming Hu, my colleague, and Yuan Li, did these studies. And essentially, we worked with GVAX, which is a GM-CSF secreting whole cell vaccine, for many years, and performed clinical trials in lung cancer and prostate cancer with that vaccine, and have been really interested about the preclinical data. This shows you that that vaccine works compared to control, but that this autophagosome vaccine worked better. So this is a set of data that was put out by Brennan in Maine, and it's the data that I almost went to their lab, actually, with his lab as a postdoc, because I was so excited by these data. And it's really the reason that I spent probably 25 years doing autologous tumor cell vaccines, or TIL, or tumor vaccine draining lymph nodes. And essentially what it is, if you take 10 identical mice, inject them all with 3-methylclanthrin, which is a carcinogen similar to cigarette smoke. And what they'll do is they'll all develop sarcomas. And if you have a pathologist look at it, they're all going to look exactly the same. If you look at gene expression profiling, the vast majority of genes are all going to be the same. But they have unique mutations that if you now take and vaccinate 100 mice with tumor A and come back and challenge 10 of them with living tumor A, you find out that they reject it. But if you inject them with any of the other sarcomas, the vast majority of mice don't reject them. So this is the reason that many people have focused on patient-specific therapies and looking at especially the neoantigen work. And this has been repeated, more than 100 times in lots of peer-reviewed literature using lots of different methods. So this PhD student, Christopher Twitty, went on to do some studies. He thought about the short-lived protein concept. He thought if that was true, we should be able to use the short-lived vaccine to actually vaccinate animals and change that paradigm. So he went ahead and did that. We developed these tumors in my lab. These are methylclanthrin-induced tumors. Essentially, this is just repeating the work of Brennan Maine. You vaccinate with 304, protects from 304, but not from these others. These X's are experiments that he did not do. When he did it now with the autophagosome vaccine, he saw a very different picture. Now if you vaccinate with an autophagosome vaccine for 304, you get protection from 304, not from 309, but you also get it for 310 and 311. And you get the protection for every other crisscross we have. So 8 of the 9 crisscrosses, we got the protection. So the reason these are black is because they were 95 to 100% protection. These are 60 to 80 and 20 to 40, but all statistically significant, and all this is published. We went on to show that the T cells were actually primed and were making interferon gamma, and they could recognize all those different tumors where they could protect. But they weren't as strong, clearly, as the whole tumor vaccine. So how are you going to increase that? You're going to increase that, I think, by using some other immunological intervention, like co-stimulatory agents. So we have a whole cohort of these different activating receptors, most of which have been tested in the clinic in combination with anti-PD-1 and have essentially failed. These are things that most companies are dropping out of. But I think it's because they've been developed the wrong way. So it turns out that if you trigger a T cell with an antigen, they upregulate OX40. It's not normally, the effector cells don't express it typically, but it's upregulated within 24 hours. So it's only there for a period of 24 to maybe 96 hours, then it goes away. That's when you should be giving the OX40 or the GITR or the CD27 stimulation. So if you think of it, it's like this. It's like if you hit the gas pedal and the car isn't turned on, it's not going to go anywhere. It's just like the same thing with taking the brakes off the car if you don't have the car turned on and moving. So the idea is you need to do something, and I'm talking about a vaccine here, but you could be using, maybe there's components for chemotherapy or radiation or other things you might be able to interject. But these two guys, again, Chris Twitty and Sean Jensen in the lab went and did a series of experiments across as a therapy model in 3 different strains of mice, so all H2 different, right? So a white mouse, a brown mouse, and a black mouse, just for quickness. They're all mammary cancer models. And what we did is we did mix and matches in terms of the vaccines to see if an allogeneic vaccine made with the short-lived proteins could actually provide now therapy, not protection, therapy in a model of 5 to 9 day tumor. And essentially, this work, I'm just going to skip to this part. Part of this was published in scientific reports, but it just showed that essentially you could get significant therapeutic efficacy by taking the vaccine. The vaccine is about here, OX40 alone is about here, but the combination gave you a much better, much more significant response. So we wanted to move that to the clinic, and that's the concept of the clinic, what I got right here, so the concept. So we've actually made this vaccine that was funded by SBIR mechanisms, but essentially we've got the vaccine. I won't spend a lot of time going into it given time. But I'll just tell you that for the clinical trial that we ran, the vaccine by mass spec has about 363 proteins in it for genes that are overexpressed by the TCGA dataset in lung adeno or in lung squamous. This was a trial that was a randomized multi-center phase 2 study that was initially led by Walter Erba, but then Rachel Sanborn took it over. And so the primary endpoint of this clinical trial was to assess immune responses. And this was done in patients as an adjuvant clinical trial. So these are patients who have had stage 3AB lung cancer. They got their definitive treatment. There's nothing else to do for them, but you're going to wait and see, and 70% of them are going to develop their lung cancer in some number of years. So the primary assessment we used was humoral, so antibody responses against proteins. The reason we did this is for a number of reasons, but really it was some work out of Larry Fong's group. This is the Serena Queck paper out of UCSF a number of years ago. But it showed that antibody responses coordinated with CD8 responses or T-cell responses. And so what you do is if you look at the antigen getting its way within the dendritic cell to the lymph node, the DC, is presenting antigen to B cells and to CD4 cells. For this B cell to switch from an IgM to an IgG, it needs to have CD4 help. The virus literature shows you that overall, by and far, is antigen specific, is the same antigen, not the same epitope that the CD4 cells see as the B cell sees. And what has been shown recently, and again, I'm not going to do it for time, but this Tripathy paper, actually which published in PNAS, shows it's very provocative because it shows in patients with lung cancer that they've got antibody responses to proteins, which if you elute from the peptidome of non-small cell lung cancer, which I would suggest are the important epitopes to be looking at, they have antibody responses against the proteins that have their peptide on the surface. And not that the antibody is going to see that, but essentially it correlated with CD8 T cell responses in 5 of the antigens or epitopes that they looked at, all of which were shared non-mutated epitopes and allowed the patient's T cells to recognize partially HLA-matched non-small cell lung cancer cell lines. So we looked at that in these patients, and essentially all the patients made B cell responses. They also had, using the adaptive T cell receptors, we saw increases that were similar to what we had seen with anti-CTLA-4, which was known to expand T cell clones. I'm just going to take you through one patient and get to the next wave clinical trial. This is looking at a patient. These first 3 time points, this is baseline day 1, day 2. I put those in there because seromics has been notoriously poorly done, right? It's very difficult to get reproducible work. But I'm just showing you that in these 3 time points, the patients are all at 1 or 1.2. But now at 3-week intervals here, you can see them going from green to yellow to red with increased antibody responses. So what are they making the antibody responses against? These are the gene names for those proteins. This is one TSHC1. It's amplified in 3% of patients. But if patients have got an amplification or upregulation of the TSHC1, they do statistically significantly worse in the TCGA datasets than the total population. And the same is true for these other proteins, other genes. And we've used that to identify T-cell responses against the same epitope. Now, one of the most interesting things, I think, for us, because we could do this assay now initially for 5,000 proteins, we could do it serially, we could watch the immune responses. And even though patients were getting vaccinated every 3 weeks, this one made an antibody response to green protein, and it came back down again, even though they were getting booster vaccinations. So it was just this concept that the immune responses were going up, but they weren't being sustained or further augmented. What happened to the T-cell responses? We were seeing the same sorts of things. That here the CD4 responses were going up. This is the same kind of 11-week time point. And then they come crashing down. CD8s were a little bit different. How are you going to change that? OX40. Co-stimulatory agents, we know, can do things to augment responses, but also blunt contractions. So instead of having this kind of response, you can get a response that goes higher. So essentially, the concept is, and I'm wrapping this up here now because I'm 20 seconds into the flashing red, the cancer vaccine turns it on, we think. The OX40 should be able, and GITR should be able to give it gas, and then we should be able to take off the brakes and amplify the response. So we have 2 clinical trials that we hope are going to launch in the next 3 to 6 months. One is an advanced triple-negative breast cancer, which will be led by David Page. This vaccine is going in with anti-OX40 and a checkpoint blocker. And we have another one, which is in advanced head and neck cancer. And again, both of these have got somewhere in the order of 300 or so proteins in the vaccine for genes that are overexpressed in that cohort of patients. So what I've kind of told you about is that, from my perspective, the mechanism of complete tumor rejection or cure is unknown, but I hypothesize it requires coordinated innate and adaptive immunity, and I take that from the IL-2 data. Reviewed the FDA objectives. I discussed cancer heterogeneity and the role for non-mutated antigens overexpressed by cancer. And then I presented you some of the preclinical data on the autophagosome vaccine products, and then where we are with the clinical autophagosome vaccine trials. The work that I presented is, I've tried to mention people as I went along. I just mentioned that my clinical colleagues, Walter Erba, who directs the Institute, without his support, none of this would be possible. I mentioned Rachel Sanborn. David Page is leading the breast cancer study, and Ron Leidner will lead the head and neck. All the antibody data I talked about was a collaboration with Fridtjof Len Johansson, who's at University of Oslo. And then there's been other things and other collaborators I've mentioned. I'll stop there and take any questions. Thanks.
Okay, we have time for one or two questions. Bernie, I'd like to maybe kick it off by asking you, you showed proximity between PD-1 and PD-L1 as spatial proximity as being a possible prognostic marker. But PD-L1 is also expressed on other cell types, not just on tumor cells, like tumor-associated macrophages. So I'm wondering whether you would get better specificity or prediction if you also specified PD-L1 on cancer cells vis-a-vis their proximity to PD-1.
So in those studies, they're looking at both tumor and macrophage expressed PD-L1. And we'll say we've got some beautiful pictures I could show you of melanoma, where the melanoma's tumor cells don't express any PD-L1, but there's a ring of macrophages around it, and they're just strikingly PD-L1 hot, right? So you see all these different, as you look at it, you see all these different things. But I agree with you. I think PD-L1, the only place we don't look at PD-L1 expression in our hands right now is PD-L1 that's expressed on lymphocytes. But we take macrophages or tumor cells as being ones that we look at. Phil.
So Bernie, I'm curious with the autophagosome vaccine, are you looking at the induction of CD4 as well as CD8 cells and trying to characterize the breadth of TCRs that are getting activated by the vaccine?
We are. So our next step is trying to look at using the hashing system in 10x Genomics to look at the follow-up biopsies on patients to compare the CD39, 4-1BB, PD-1 positive cells that we get in the biopsy before treatment and after treatment, how it changes. And so what we've been doing so far is trying to characterize what we see in terms of the adaptive technology, but with the 10x Genomics, we get both the alpha and beta chains. We reported at SITC this year, we've already pulled out from patients that have gotten the vaccine, we've been able to pull out alpha beta T cell receptors. We can actually put those into PBMCs and they recognize the patient's tumor. We're working on a collaboration now to try and use a more high throughput way of screening those to see what they recognize. But I think that's going to, my own perspective is that's going to add such a great dimension. I mean, the data you showed was amazing, right? You can now look into patients that are responders or non-responders and what their gene profiles. But we're very excited about using that and applying that to our patients.
And have you looked at whether or not it's also inducing FOXP3 positive CD4 cells?
Yeah, we're trying to look at that. We'll hopefully see that when we start to do these biopsies. But we've not, I don't have any, we haven't really looked at it yet.
One more question, please.
Okay, following up on Elia's question, did you look into the spatial profiles of these different T-cell populations that change upon therapy? For example, do the spatial profiles change? And then is that diagnostic or predictive in some fashion?
I'm sorry, the patient, what has changed?
So you talked about the spatial profiles.
Yeah.
So have you followed them over time, and then do the profiles change just in terms of spatial locations, and is that predictive?
So I would just say that the studies we've done so far in the clinic have been in the adjuvant setting, so we don't have access to biopsying tumor because we didn't think that vaccine alone was an appropriate therapy to give to patients since vaccines alone don't work. And so in that setting, so in this current study where we will have biopsy pre-treatment, and there's mandated on the head and neck 8 and 12-week biopsies. If we can get that kind of data, we will actually be able to look at that much more closely. Thank you. Great question.
Thank you.
Thank you.